Hello, and welcome to the final podcast for Chapter 20, Day 20. In this podcast, we're going to cover Objective 2. And in this objective, we're going to talk about organismal cloning. We're going to talk about the differences between the different kinds of stem cells. And then we'll also talk a little bit about how Dolly, the first mammal that was cloned, how that was done. All right, let's go and get started on this important topic. Okay, as we begin this podcast, I think it's important that we start with the difference between embryonic and adult stem cells. These two terms are used quite a bit in the media, and in some cases they're used incorrectly. So let's go ahead and start off with a good understanding of these two terms. And the first thing I want to tell you is what are stem cells? Just the generic term stem cells. Let's think about what that means. So stem cells are important because, well, let's draw. This is a generic stem cell. We know we need these during embryonic development and fetal development for growth and differentiation. This is really important. We all start off in this world as a single cell, and to this day, this is one of the biggest things in biology that just blows my mind, that you have this single cell and it has all the genetic information in it to begin to divide and to differentiate to become a living, walking human being, right? That is fully differentiated and has all the characteristics and all the physiological processes that we all have. And it all starts in this one single cell. It totally blows my mind, right? Everything in this stem cell here is what we need. It's got the genes we need. It has the transcription factors that are necessary to turn on the genes necessary to begin this process. It's a very cool phenomenon. And one, like I said, that still kind of freaks me out a little bit. It's pretty amazing. It's the reason I got interested in cell and molecular biology, thinking about these single cell zygote. Another purpose of stem cells is for repair. So we have stem cells all over our body right now as adults. And they're very important in repairing damaged tissue that's been destroyed because of, say, a disease or destroyed because of a trauma. Stem cells are really important in repairing those cells. They're also important in kind of going with repair, but I'll put it under here too. Well, let's just make another arrow. They're important in replacement. We're not born with all the cells we're going to need for the rest of our lives. Every cell has a finite life. It divides a very specific number of times and then it dies. And it's really important it does that, because if it doesn't, then it starts accumulating mutations, and then these mutations can lead to diseases like cancer. So it's really important that cells, when they reach a finite lifetime, that they die. And the only way we can replace them is using these stem cells. Okay. So these are three important aspects of stem cells. Growth and development. Primarily, we think about that during growth and differentiation. We primarily think about that during embryonic development and fetal development, but we do see some of that throughout life. Replacement is incredibly important, as I said, to replace our dead and dying cells. And repair. Sometimes our tissue gets damaged and we need to repair that by building new cells. And that comes from these stem cells. These stem cells are always there ready to replace cells that need to be replaced. All right, so let's think about a stem cell. This stem cell, when you look at it, it looks pretty much, all stem cells look pretty much the same. They are circular. They don't appear like any specialized cells. But the DNA in this stem cell, regardless if you're talking about an embryonic or a adult stem cell, it has the DNA in it to allow it to become almost any kind of cell there is. It just has to be told to become a neuron, to become a muscle cell, to become a pancreatic cell, to become an eye cell. And then it'll do that. Okay, now what I want to do is really focus on some of the differences between embryonic and adult stem cells. Okay, in this slide, let's talk about the different types of stem cells. On the last slide, we talked about the general nature of stem cells. Now let's break it up between embryonic stem cells and somatic stem cells. Okay, in this slide, we see 6 pictures here, and each of these pictures represent a stage of human development. Each of these pictures also represent a source of stem cells. So first, let's label what these stages of development are. We have a zygote, a single-celled fertilized egg, and then we have a morula stage. This consists of 8 cells so it's after 3 divisions. And over here we have a blastocyst. This blastocyst consists of, well, essentially it's this ball of cells here. On the outside are cells that will go on to become um, what we call extra embryonic tissue, such as the placenta. In the inside, we have the inner cell mass. It's called this because it's on the inside of this embryonic stage, and it's a large mass of cells. This is what will go on to become the body of the embryo and the fetus, and ultimately the child and adult. Then down here we have a fetus, about 12 to 15 weeks old here. Here we have a child, a rather grumpy child for some reason. And here we have an adult. As I said before, each of these are a source of stem cells, so let's identify what they are. In this top row here, these are all embryonic stem cells. So I'm just going to write embryonic stem cells across the top here because that's what all of these are. On the next slide, we'll break these down into more specialized terms. And down here, these are called somatic stem cells. Somatic stem cells are found in an embryo stage, which isn't shown here. Once tissues have started to form, we call that's when the stem cells lose their embryonic stem cell nature and become somatic stem cells. So we even see it before this fetal stage. As we talk about somatic stem cells, it's a good idea to define what we mean by somatic. All of our cells in our body are either somatic cells or gametic cells. Somatic cells are every cell in our body except our gametes, that is, except our sperm or eggs, depending upon our gender. It's important to have these somatic stem cells because they replace all of our somatic cells when they die, either to natural causes or due to injury to those tissues. So that's what a somatic cell is, and that's why we have somatic stem cells. The reason somatic is a better term because, as you can see here, these stem cells are not only found in our adult here, they're found in a fetus and they're found in children. So adult stem cells is not the most descriptive term. While we're on this slide, it's also important just to mention real quick that it is this stage here, the morula stage, and the blastocyst stage. These are the sources of embryonic stem cells that are used for research. These are obtained from in vitro fertilization clinics when the parents have decided to donate these after they have decided they're not going to use them anymore. They can voluntarily donate these to research. Okay, so let's move on to the next slide. In this next slide, we're going to talk about the specialized terms we're going to use to describe embryonic stem cells, and then we're going to go on and talk about how embryonic stem cells and adult stem cells can be used in research. Okay, so let's go ahead and differentiate the different kinds of embryonic stem cells. So over here we have our zygote, our fertilized egg, and then it will divide to this 8-cell stage. I'm going to do my best to try to draw 8 cells here. And we call this the morula stage. Then it will continue to divide till it gets into this more specialized blastocyst stage. I'm just going to draw it like this with our inner cell mass here. So it looks like this big mass of cells here, which will eventually become the body of the embryo and fetus. And around here are other cells that start to form and differentiate, and these become like the placenta, the amnion, another term called the atlantois cells. These are extra embryonic membranes, and they're very important for the development of it. but only this inner cell mass becomes the actual embryo. And then it becomes developing fetus. And then within this fetus, as it's growing, and certainly by the time it's born, um, we will have many different kinds of tissue forming. We'll have heart tissue, we'll have the immune system, the digestive system. They have become specialized in this at this stage here. Okay, oh, let me write blastocyst. I didn't put that here. So the morula stage here and the blastocyst stage, these are both embryonic stem cells, but they're a little different. The morula stage, we call these totipotent stem cells. They're embryonic stem cells because these are part of the embryo. I have to remember this because toti sounds like total. They have total ability to become any cell in the embryo and the developing fetus. So we're going to say any body cell, meaning any of these cells could become a heart cell, a neuron, a stomach cell, what have you. And they also have a very unique ability to become these extra embryonic cells that we see in the blastocyst stage and as the fetus develops. So like the amnion, the yolk sac, the placenta, there's a few others. We're not going to ask you those questions on this test, just so you know what they are. So it can become any cell. They have a great deal of potential. Now the blastocyst, we call these pluripotent. They have a lot of potential, but not quite as much as the totipotent. They're pluripotent cells. They have the ability to become any body cell. So they can become a heart cell, a stomach cell, a big toe cell, any body cell. So that's pretty cool. Now within the fetus, as these different tissues are being formed, and certainly as the baby is born and develops into an adult, all of these tissues here are differentiated. So I'm going to write differentiated underneath it. So the heart, immune, digestive, they are differentiated cells. Now within these tissues though, so let's draw a heart here. Obviously not an anatomically correct heart, but so here's our heart. It has all the cardiac muscle cells and other supporting heart structure cells in here, making this tissue and this organ a very differentiated functional set of cells here. But within this heart tissue, as well as within the digestive system, we have specialized cells in them. So kind of mixed up here with these differentiated cells, we have stem cells. The same kind of stem cells we would see that we talked about in the last slide. And remember, we want to call these somatic stem cells. It's what a lot of people will call adult stem cells. But remember, we want to try to get away from that because it's important for us to realize that in the developing fetus, those are these somatic stem cells. It's important that these tissues have it so that they can fully differentiate and that they can begin to replace cells as they die or as they are damaged. You can imagine if you have a heart attack, how that damages a lot of these cells. Well, stem cells we know can help fix that. In fact, some really cool studies have taken stem cells from other parts of your body, like from your leg, and when they do an open heart surgery, there's damage to that heart, they place stem cells on that heart, on that damaged heart tissue, and that helps repair it. So these stem cells are really important. Now, these stem cells here, they don't have the ability to become any body cell, and they don't have certainly not the ability to become one of these extra embryonic stem cells, but they have the potential to become at least any kind of cardiac cell. So we call these multipotent cells. So again, you should know that embryonic stem cells are found in the morula stage and the blastocyst stage. They can be differentiated between totipotent stem cells in the morula stage, which have more potential, and then the blastocyst stage, which are pluripotential cells that can become any cell except for these extra embryonic ones. And then we have the somatic stem cells, which are different than the embryonic stem cells because they don't have quite as much potential to become any cell. And that's why some of the controversy we see about using adult stem cells versus embryonic stem cells, um, the different kinds of potential they may have. So now with that in mind, let's go back to our diabetes problem. If you remember, we talked about in the first objective how we could use gene cloning to mass produce insulin. And when we mass produced insulin, we could produce it more efficiently, and it turns out to be a very effective way to treat diabetes. But again, it's not a cure. Then we talked about gene therapy, and the prospect of gene therapy by replacing the mutated gene with a gene that's not mutated has a lot of interest and a lot of potential. But we are finding some problems with gene therapy that has not made that come to reality yet. But what I want to talk to you about is how scientists have been using embryonic stem cells as a potential way to cure diabetes type 1. So what I want to do here, just try to explain how this works. I'm going to draw some embryonic stem cells. I'm just going to draw them as kind of these very similar looking circles. And when a stem cell divides, it does one of two things. It either divides to make a new stem cell, because remember, we have to keep replenishing stem cells, or it specializes. And scientists know how to take these embryonic stem cells and allow them to keep replicating and forming new ones. So scientists are learning how to take these embryonic stem cells and by providing certain signals, allowing them to differentiate into specialized cells. In fact, they know how to convert these embryonic stem cells into the pancreatic specialized, fully differentiated cells that make insulin in the pancreas. So these cells will now make insulin. One of the problems with embryonic stem cells is that they are not an exact match to the person who would receive them. And so there is a concern that if you were to place embryonic stem cells in a person, our immune system would go after them. And there's good reason to think this, because our immune system will go after them because they're going to look at them as foreign cells. And there's ways to get around this. But one way that scientists are thinking about doing that is they take these pancreatic insulin producing cells that they obtain from these embryonic stem cells. In fact, why don't we write that above here? They have taken these insulin producing pancreatic cells and they have put them sort of like in this little capsule. In this little capsule, they can place it in the body, or I think they can even keep it like outside the body, like in a little pocket. And as the individual eats something with sugar in it, and they need to have insulin pumped into the blood system, this artificial pancreas, so to speak, will put pancreas will make will make insulin and secrete it out into the circulatory system. The advantage to this protects these cells against the immune system, so they're going to be in this extra kind of packet protected from the immune system. So it's a clever little trick that the researchers have come up with. In this way, a person with diabetes type 1 who doesn't produce insulin, we're giving them cells that produce insulin. We can't ignore the fact that these embryonic stem cells, using them is still quite controversial. Whatever side of this debate you are on, I think everybody can agree that there is still controversy. One way that scientists are trying to alleviate some of this controversy is to create a different kind of stem cell. Now, the stem cell I'm going to talk about, they're not as from what scientists will tell you that work in this area, they're not as good as these embryonic stem cells from the morula cells, but they're a step in the right direction. What we know we can do here is we know we can take, let me draw one of my favorite famous drawings here of a person. Now this person here, very short legs, sorry dude. We can take any cell from this person. Let's take it from their arm, and let's magnify this cell. We know this cell has a nucleus, of course, and it has 46 chromosomes in it. We know that these 46 chromosomes contain roughly 21,000, 22,000 genes. And we know that these genes are turned on or off depending upon what tissue you're in. We've known for a long time as scientists that in order to take this fully differentiated epithelial cell from our skin and to convert it to a stem cell, an embryonic stem cell, that it's just a matter of gene regulation. Of these 21, 22,000 genes, different genes need to be turned on, and then we could change the differentiation of this cell. And I would say maybe 8 years ago, I would teach students that we have to change the expression of 200 genes in order to make an embryonic stem cell. Well, that seemed unreal. We can't really change the expression of 200 genes. And scientists kept working on it. And that number got lower and lower and lower. And finally, we've gotten to a really manageable number. We know that if we take 3 genes out of these 21, 22,000 genes and overexpress them, that this skin cell will become an embryonic skin cell. I'm just stem cell. Let me say that again. Overexpressing 3 genes in this skin cell from the epithelium of this fully differentiated person here, we overexpress these 3 genes, and now it remembers, so to speak, how to become an embryonic stem cell. So how does it do that? Well, I'm going to draw these 3 genes down here. Don't worry about what their names are. That's not so important right now. But this is a good opportunity to review some of the things you should have from transcription and translation. So remember, we have our promoters here. And these promoters of these 3 genes, if we activate them by throwing on our RNA polymerase, transcribing these and making messenger RNA through transcription, and then these messenger RNAs being translated into protein, these proteins now will act to change the cell to a stem cell. Not surprisingly, if you were to take a moment, and you can hit pause if you want to think about this, what kind of genes, what kind of proteins are encoded by these 3 genes? Remember, these genes are going to tell this whole cell to become an embryonic stem cell. So the common theme that we're seeing here is that these are actually transcription factors. And you activate these 3 transcription factors, and presto changeo, you've just made a stem cell. Pretty cool. Now, these are these are called iPS cells, and that stands for induced pluripotent stem cells. So they're not quite the same as the totipotent stem cells, but they're still pretty cool. They're still pretty good. They still will differentiate into any kind of body cell. And so the hope is that maybe we can use these stem cells, these induced pluripotent stem cells, to do the same kind of stuff that we're doing with the treatments or the potential cures for things like insulin. They offer less controversy because they have not come from an embryo. They also offer the advantage of being from the person who needs them. So if I needed them to cure a disease I had, I could be confident that my body's going to recognize these as self cells, my self cells, and they're not going to be destroyed by my immune system. So they do offer some potential. Okay, so the last topic we want to talk about is how organismal cloning. And as an example, we're going to talk about how they cloned Dolly the sheep back in 1997. That's been quite a while ago, and so there's been many advances taken forward from there, but the principle hasn't changed too much. So we're going to just look at Dolly. This will probably shock you, but I am not a very good drawer of sheep. So here's a green sheep, prepared to be mesmerized. Okay, that's a lovely sheep. And let's draw a red sheep over here. Equally impressive. All right, let's say we wanted to clone this red sheep. We wanted to make an identical clone of Mr. Red Sheep here. What we do is we take from the green sheep one of her eggs. Okay, so we take one of her eggs. Let's draw her egg here. This is her oocyte. Of course, I drew it bigger. It's almost the size of a sheep, but it's much, of course, much smaller, right? And it has this nucleus inside of it with all of its DNA in it. So let's just draw like one strand of double helix here, just to say it has that DNA in there. Then what they do is they take a pipette and they remove this DNA. So you end up with a cell that looks like this now. The oocyte stays the same, the egg, and now we have our nucleus here but with no DNA in it. We just remove the DNA. Now what we do with our red sheep over here, we remove one of her epithelial cells, one of her skin cells. So this is her epithelial cell. It's a fully differentiated cell. And she has her nucleus here and also with DNA in here. And what they do here is they take another pipette and they remove this DNA. They don't throw it away. They put it in a little tube here and keep it set aside for just a moment. So in this tube here we have all of her nuclear DNA from this one cell. Then we take this DNA here and we place it in this egg here. So this is the green sheep's oocyte. And we place the red sheep's DNA in here. All chromosome sets, so all 46 chromosomes are right in here. And now we induce, or they induce, this cell to begin to divide. And what happens here, it still kind of freaks me out a little bit what happens here. It's pretty cool, is that when it starts to divide, it divides like it's a zygote. It kind of remembers, if a cell can remember, it remembers what it's like to be a zygote. It immediately turns on the transcription factors, or I should say, within the cytoplasm of this egg, there are already transcription factors that now begin to bind to this DNA that just came from this red sheep and tells it, now you're going to be programmed to be a zygote. And now it divides and grows, and then they take this egg and they place it back into the green sheep. Okay? And they let it develop, and when it gives birth to a sheep, it gives birth to a genetic clone of the red sheep. The reason it's a genetic clone of the red sheep is because it has the DNA from the red sheep. So it's got to be an exact clone of it now. So this is how we, in general, will clone an organism. We'll take the DNA from one organism and place it in an egg of another, and then place it back into the uterus and allow it to fully develop and differentiate. And we end up with a genetic clone. This is actually done, I think people might be surprised to know this, but this is done a lot, particularly in the agricultural sciences. Various types of agricultural products, even from like cows and horses, are cloned like this, or can be cloned this way. Okay, well that ends this podcast and this chapter. If you have any questions over it, please let me know, and we'll do our best to answer any questions you might have. We'll see you in class. Bye.